Hello, my name is Dylan Shiner, and for my project I focused on the effects of a forward osmotic and antimicrobial fiber membrane made of these two polymers, polyvinylidene and polyaniline, and their effects on gram-positive and gram-negative bacteria in wound sites. The first thing I'd like to go over is the motivation and the state of the art of the project. So a little bit more about the motivation is that wound site infections can be very dangerous due to bacterial colonization on them. Approximately 15 million deaths a year are due to wound site infections, and bacteria are becoming more resistant to antibiotics due to things like biofilms. So as you can see in this diagram, a little bit of bacteria attaches and then it grows to form a little colony, and then the colony gets a little bit bigger until it really settles in and forms this biofilm, which isn't really penetrable by antibiotics. Staphylococcus aureus is a prime example of a deadly disease that's resistant to this. It goes through this exact process of forming this biofilm that can't really be treated by antibiotics, so new methods have to be formulated in order to combat this adaptation that bacteria has now created. So to go into the state of the art of what we'll be using for the project, electrospun materials are kind of what will be used to create this fibrous mat for polyvinylidene and polyaniline. So there's two methods to creating electrospun membranes or electrospun materials. One method is electrospraying and one is electrospinning. What we have in the diagram right here is electrospinning. Electrospraying, you just spray the material down onto like a random mat, and it just creates a arbitrary shape. There's no like control to it. So we use electrospinning because we can make a kind of solenoid of the materials, and then we can then lay it out to create a fibrous mat that can be kind of weaved together. So mostly polymers are used, which is why I use polyvinylidene and polyaniline to make these resulting fibers. So the process of creating these using like this diagram is that there's a metallic needle and a voltage is applied to it, and this voltage creates a charge inside the polymers, which then creates a kind of droplet that projects outwards towards the ground source, which is this rotating collector right here. So now we'll move on to the background. So now what comes into question is, why did I choose electrospinning, and then why did I use polyvinylidene and polyaniline as the base for the fibers for electrospinning? Well, I chose electrospinning because it's a new process that's fairly cheap to do, and I chose polyaniline because it's a polymer that's naturally conductive. So when you add a dopant like chloroxanol to the mixture, the polyaniline becomes positively charged, which can displace ions within cells and bacteria. So the positive charge from the membrane displaces calcium ions and magnesium ions within the bacteria, or generally any positive ions, doesn't have to be those two specifically, and it causes a penetration of ions in and out of this bacteria cell. I chose polyvinylidene as the other polymer to go along with polyaniline because there's no driving force attracting the bacteria to the mat or the gauze to be killed. So basically, when polyvinylidene is coated with polyvinyl alcohol, it increases the hydrophilicity by a substantial amount, which in turn increases the effect of forward osmosis, which uses the osmotic pressure gradient as the main force of driving water in and out of the membrane from one side to the other. And this biofouling effect will occur where on the surface of the membrane right here, bacteria will start to accumulate, and in turn, because of the polyaniline being there, they will be killed, which will drive the bacteria away from the wound site and also kill it, preventing any colonies from being formed. So the next part to go into is the strategy and approach. For the first part of the strategy, we kind of need to find out the proper composition and structure of the fibrous mats in order to kill bacteria in the most efficient way possible. So there's two configurations we can go about creating the fibrous mats. One is like a layered structure where you have like polyaniline first, and then the next part of it is polyvinylidene, or it can be vice versa. And then the other configuration we have is a mixtures mat where it's just, there's no really layered structure to it. It's just kind of polyvinylidene, polyaniline are in the mat in set compositions, but there's no structure to it. They're just kind of there. So the next part is to create set compositions of these two configurations. So polyvinylidene will be at 30%, while polyaniline is at 70%, and then it will go to 40-60, 50-50 until you get to 70-30 by composition and weight. Once these fibrous mats are created, they will be placed in CFU assays, which have either gram-positive or gram-negative bacteria, which will be tested to see how efficient each of these samples can kill bacteria. The colony count for this part will be measured every hour once the sample is introduced until around 12 hours. So it's around 12 data points. And then the three best samples will be taken for further testing in the next steps of the strategy. So the next step is doing tensile testing. It has to be sturdy enough for it to not tear while being applied to a wound, and it has to be able to continuously go through wear and tear while on the wound. So tensile testing, stress and strain tests will be done for the three most efficient mixtures to ensure their practicality and make sure that they should be applied to wounds instead of them just breaking, I don't know, every minute once they're applied. The third and final part for the strategy, or the experiment in this case, is to test the fibrous mats on animals to see if they can treat real wounds and put them in a simulated environment to see how effective they are as opposed to in assays. So once a wound is created, gram-positive or gram-negative bacteria will be introduced to that wound. No extra procedures will be done to treat the wound. 10 mice will be used for each sample to just get a varying amount of data points. So after 1 hour, 6 hours, 12 hours, and 24 hours, samples will be extracted from the wounds and tested to see how efficient the fibrous membranes are at killing bacteria on actual wounds as opposed to in the assay as what we did before. So just what I want to get out of the entire experiment and the project is that it's becoming more difficult to kill bacteria overall because of this adaptation to create a biofilm where antibiotics aren't able to penetrate and kill bacteria. So we create a different method for killing bacteria, which has to do with the ions within the bacteria themselves before they can create this biofilm, ideally, which displaces the positive ions within the bacteria and kills them before anything serious can happen. And also we want to take the bacteria away from the wound before it gets there, so that's why polyvinylidene is used to prevent them from sticking to the skin, and they'd rather stick to the fibrous mat that we've created. And the last part is just the experiment to ensure that the structures are the most efficient way they can be made, and the compositions are the most efficient way they can be made, and that there's a proper creation of the fibrous mats that don't wear and tear and break very easily. So in all, electrospun materials are just a very efficient way to kill bacteria in a new method that doesn't need antibiotics. Now I'd like to thank everyone who watched this for their attention on my project, and if you have any questions, feel free to ask them now.